Rahmatullahi. These days we are working on the chemical basis of life. In the last class, I have already started to discuss the nucleic acids. We are here. Nitrogen bases, these things I have already explained you. Right. So, in today's lesson, we are introducing ribonucleotides. Look at this diagram. How many ribonucleotides are there? Mean there are 4 ribonucleotides are there. Actually, these 4 ribonucleotides are different from each other due to their nitrogen bases. Okay, this is adenine nitrogen base. This is guanine nitrogen base. This is uracil nitrogen base. This is thymine nitrogen base. So all together, not thymine, cytosine, sorry. So how many ribonucleotides?
4 types.
4 ribonucleotides. In all the 4 ribonucleotides, there will be ribose sugar. There will be phosphate group. Okay. Ribose sugar, phosphate group. Ribose sugar, phosphate group. Ribose sugar, phosphate group. Ribose sugar, phosphate group. That is same. Which component differs? Which component differs? From one ribonucleotide to another ribonucleotide, which component differs?
The nucleotide?
Yeah, one nucleotide and another nucleotide is different in this nucleotide and this nucleotide and this nucleotide, they differ in which feature?
By nitrogen bases?
By nitrogen bases. By nitrogen bases. That is very important, by nitrogen bases. So there are 4 nucleotides. Ribonucleotides are how many? Ribonucleotides are how many?
4 types.
How many ribonucleotides are there? 4. Those 4 are different from each other in their type of the nitrogen base. Are you clear?
Yes, sir.
Yes. Coming to the next one. This is deoxyribonucleotides. Here the sugar is deoxyribose. Here this sugar is what kind of sugar? What kind of sugar? What is the sugar there?
Pentose sugar.
What is the sugar?
Deoxyribose sugar.
Deoxyribose sugar. Now see, in this place, can you see an OH? Can
Yes,
you
sir.
see? In this place, what do you have?
Hydrogen.
Hydrogen. O is gone, correct?
Yes.
That is how we have to identify ribose and deoxyribose. In this place, if there is OH there, then it is
Ribose
ribose.
sugar.
If it is only hydrogen there, deoxyribose sugar. So here this is adenine, this is guanine, this is thymine, this is cytosine. How many deoxyribonucleotides are there?
4 types.
4. They are different also according to the nitrogen
Bases.
base. Clear?
Yes, sir.
Right. Remember, none of these structures you have to memorize. It is not my expectation. Understood? It is not your examiner's expectation. Don't try to memorize these structures or these structures. Clear?
Clear, sir.
Right. So don't memorize. Just to tell you, there are how many nucleotides in the ribonucleotides? How many are there? How many ribonucleotides are there?
4.
4. Those 4 are different from each other in which feature?
The nitrogen bases.
The nitrogen bases. What are the nitrogen bases possible?
Adenine, guanine, uracil, cytosine.
Cytosine. Similarly, deoxyribonucleotides are how many?
4 types.
4 types. They also differ from each other in which feature?
The nitrogen bases.
The nitrogen bases. What are the nitrogen bases possible?
Deoxyadenosine, deoxyguanosine.
No. The nitrogen bases possible. That's all I asked.
Adenine, guanine, thymine, cytosine.
Don't try to read what is there. Okay. Understood?
Yes.
Right. Remember, before we further go into, now we are going into ribonucleotide and what is it? Deoxyribonucleotide. You know, ribonucleotide mean what is the sugar there?
Ribose sugar.
Ribose sugar. Deoxyribonucleotide mean what is the sugar there? Deoxyribose sugar. Ribonucleotides are making the nucleic acid called RNA. Deoxyribonucleotides are making the nucleic acid called DNA. In the ribonucleotides, uracil can be seen. Will there be uracil in the deoxyribonucleotides?
No.
No. To that place only who is there?
Thymine.
Thymine is there. Clear?
Yes, sir.
Good. Before we come to this part, remember, when a pentose sugar, it can be either ribose or, what is it? Deoxyribose. When it is connected to the nitrogen base. Now, who two are connected?
Pentose sugar and nitrogen.
Pentose sugar and nitrogen base. The product is called nucleoside. Understood?
Yes, sir.
Pentose sugar and nitrogen base is called, what is it? Nucleoside. Nucleoside, when connected to the phosphate group, now it is called nucleotide. Clear?
Yes, sir.
So, pentose sugar connected to nitrogen base, product is called
Nucleoside.
nucleoside. Nucleoside and phosphate group connected mean now the product is called
Nucleotide.
nucleotide. Could you please take a white sheet and mark it down? Completed?
Yes, sir.
Good.
Yes.
Here, look at this diagram. This is a nucleotide. Why do you say it's a nucleotide? Pentose sugar is there, nitrogenous base is there, phosphate group is there. Correct?
Correct, sir.
If all these three are there, how do you call it? nucleotide. In this nucleotide, if you take this part alone, what is it?
Nucleoside.
Nucleoside. Clear?
Clear, sir.
I want you to draw this diagram and mark or put a circle and mark that is nucleoside. Can you do it?
Okay, sir.
Tell me after completing it.
Done, sir.
Good. So, hope you can understand the difference between nucleotide and nucleoside. In exam paper even, they can ask you to, they might give you the diagram and ask you to put a circle. Clear? Are you clear?
Yes, sir.
Clear, sir.
Good. So, here, what is the heading for you? Nucleotides in living cells other than those found in the home.
Nucleic acid.
Nucleic acids other than those found in the nucleic acid. Okay, I will explain you this one further. First of all, just try to grip the nucleotides there. I think out of these 1, 2, 3, 4, 5, I think you are familiar to ATP, correct? Are you familiar to ATP?
Yes, sir.
Yes. Adenosine triphosphate. Okay. Like ATP, okay, there's another component called ADP, adenosine diphosphate. Remember, okay. Now, next one, how do you read it? NAD plus. How do you read it? How do you read it?
NAD plus.
NAD plus. Okay. And next one, FAD. Other one, NADP plus. Understood?
Understood.
The point it is, their expansions are not in syllabus. Their expansions are not in syllabus. If you like, I will tell you. NAD mean nicotinamide adenosine dinucleotide. Nicotinamide adenosine dinucleotide. FAD means flavin adenosine dinucleotide. NADP plus mean nicotinamide adenosine dinucleotide phosphate. But do you want to know these words in expanded form mean 100% not necessary? So you want me to give you or skip it? What do you say? What do you say? You want me to give you or otherwise you want me to skip it? Tell me.
No need, sir.
Good. Keep that attitude. Understood? Keep that attitude. Some children are there, they always think, what is it? Doesn't matter, give sir. Doesn't matter, give sir. If it is not necessary, don't try to take it into your heads. Understood? There are so many things to study. There are so many things to work. So if we keep our brain the space only, when many work things are flowing into our brain will be having space. If we put unnecessary things inside our brain, ultimately brain will be stucking. Right. Anyway, I will give 1 point to you. This is a dinucleotide. What kind of component? What kind of component?
Dinucleotide.
That means how many nucleotides have joined?
2
Two nucleotides. NAD plus is a dinucleotide. FAD is a dinucleotide. NADP plus is also a dinucleotide. If you put all these three bracketed and say dinucleotide also, I am okay. Clear?
Yes, sir.
Have you marked it?
Yes, sir.
And keep in the mind, you can mark it down here. Later we will mark it here. It's already marked to you. Who are single nucleotides? ATP and ADP. Who are dinucleotides? These three. Please do one thing. Cancel this. Okay. cancel this middle word that is not in syllabus. FMN is not in syllabus. So all these molecules have adenine in their structure. That's why there is an A there. What is the common letter found in all these? What is the common letter found in all these? ATP, ADP, NAD, NADP, FAD. What is the common word you find in all of these? A. That A is standing for which nitrogen base?
Adenine.
Adenine. That's what you want to know. Clear?
Yes, sir.
Right. So what is the function of ATP? What is the function given? Works as an energy carrier. Cut this one and say carrier. Take your Tipexen. Take your Tipexen. Remove this and say energy carrier. Enough. It's not a coenzyme. It's an energy carrier. Works as energy carrier. Please Tipex it and write. What is it? Energy carrier. Okay. Corrected it.
Done, sir.
Right. Here it is given another name, Baba. It is called universal energy carrier. What is the other name given for it? What is the other name given for it?
Universal energy carrier.
Universal energy carrier. Why do we say universal? Mean in the entire biological world, if you take a bacteria, fungus, to the most complex organism, human, in all the organisms, who is the energy carrier?
ATP.
ATP is the energy carrier. That's why it is called universal energy carrier. Clear?
Yes, sir.
Right. So is ATP a coenzyme? Is ATP a coenzyme?
No.
No. Okay. It is not a coenzyme. It's an energy carrier. Universal energy carrier. Are you clear?
Yes, sir.
Right. Coming to the second one. Who is the molecule? Who is the molecule? NAD plus. Okay. NAD plus is described to you as a coenzyme. Okay. It's a coenzyme, mean an enzyme's friend. Without that friend, enzyme cannot do its work. Understood? Understood?
Yes, sir.
We will later, inshallah, when we study about enzymes, we will fully work on that heading. Okay. Here, this is an important point. there is a vitamin related to this. That capital N is standing for that vitamin. What is the vitamin? Niacin. NAD plus is related to which vitamin? Which vitamin?
Niacin.
Niacin. Vitamin B3. What is the vitamin?
Vitamin B3.
Vitamin B3. In the brackets, mark it down. Vitamin B3. Okay.
Okay, sir.
Right. Works as a, here you have a small correction. Okay. Exceptor words are removed. Okay. You have to use them as home. You have to use them as home. What kind of molecules they are?
Carrier.
Electron carriers. Electron carriers. Correct those two words, except the two words are there. They are electron carriers. Okay.
Okay, sir.
Right. So finally, NAD plus is working as an oxidizing agent during cellular respiration. When you study cellular respiration, you will fully understand the meaning of that word. At the moment, just pick it up. Okay. At the moment, just pick it up. When you study cellular respiration, you will fully understand the role of NAD+. Clear? Clear?
Yes, sir.
Good. You come to the next heading, FAD. FAD, flavin adenosine dinucleotide. It is also an energy carrier. It is also acting as a coenzyme. The vitamin related to FAD is whom? The vitamin related to FAD is whom?
Riboflavin.
Riboflavin. NADP plus. NADP plus is also a coenzyme. Cofactor okay, Baba. Cofactor okay. Bracket you mark it down. Coenzyme. Okay. Works as a cofactor. In brackets mark it down. Coenzyme. So who are the coenzymes here? Who are the coenzymes here?
NAD plus, FAD,
NAD plus, FAD, and NADP plus. Who are the electron carriers here?
NAD plus, FAD, NADP plus.
Right. In this one, you have one more point to mark. That is, it is acting as an oxidizing agent, okay, in photosynthesis. Acting as an oxidizing agent in whom?
Photosynthesis.
Photosynthesis. Who is the oxidizing agent during cellular respiration? Who is
NAD
the
plus.
oxidizing agent in cellular respiration? NAD plus. Who is the one in the photosynthesis?
NADP.
NADP plus. Have you marked the details?
Yes, sir.
Right. So let's go for a small summary before we go ahead. That is my usuality. Okay. Electron carrier nucleotides. Who are the
NAD plus, FAD,
NAD
NADP
plus,
plus.
FAD,
NADP plus.
NADP plus. Okay. Coenzyme nucleotides.
NAD plus.
Hmm.
NADP plus.
FAD, correct? NADP plus. Oxidizing agent nucleotides.
NAD plus, NADP plus.
NAD plus, NADP plus. Correct?
Correct, sir.
Right. Further, I will mark respiration related nucleotides. Respiration related, what is it? Nucleotides. In the respiration, my children, first, who will be contributing?
ATP.
Hello. 8. Good morning. எந்த கிரேட் அவங்க? நீங்க வந்து அவங்களோட பேரை போட்டு சரியா எனக்கு வாட்ஸ்அப்ல மெசேஜ்ல போட்டு விடுங்க சரியா நான் உங்களுக்கு டீட்டெயிலாம் அனுப்புறேன். ஓகே ஓகே. Right. Have you marked these details? And another one, photosynthesis related nucleotides. Photosynthesis related nucleotides. They are one ATP, second NADP plus. Please mark down these details. Have you marked it?
Yes, sir.
Good.
Yes, sir.
Are you clear? Remember, these kind of summaries can come in the exam. Clear? They are very, 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 very important. Good. Now, getting back to the main tute, what is the heading for you? What is the heading for you? Deoxyribonucleic acid, shortly called, what is it?
DNA.
DNA. If you take a DNA molecule, normally, I think you are already familiar this kind of an arrangement of DNA molecule. Yes or no?
Yes, sir.
Yes. This shape is called Okay. Double helix model. What is the name given to this? What is the name given to this?
Double helix model.
Double helix model. That was introduced by two individuals. One it is Watson, other one is Crick. That's why it is also called by the name Watson and Crick model. Double helix model has another name called, what is it? Watson and Crick model. Clear? Please
Yes.
mark it down. Please mark it down. Okay. Have you marked it?
Done, sir.
Right. In a DNA molecule, there will be two polynucleotide chains. I am now taking this polynucleotide chain. Okay, on the other side, there is another polynucleotide chain. Okay, now my children, if you take this one, it is the third carbon of this nitrogen, pentose sugar. Which carbon? Which carbon?
Third carbon.
This is the which carbon?
Fifth carbon.
Fifth carbon. Okay. So here it will be the fifth carbon. So again, this is, what is it? Third carbon. Here there will be which carbon? Fifth carbon. Here there will be which carbon? Third carbon. Here there will be which carbon? Fifth carbon. Like that, if you go, in this end, there is a 3 carbon, correct? Am I right?
Yes, sir.
Right. This 3 carbon doesn't involve in making phosphodiester bond. Understood? Here there is a phosphodiester bond. Here there is a phosphodiester bond. Here there is a phosphodiester bond. Okay. Who are the carbon atoms involving in making the phosphodiester bond?
Fifth and third.
Fifth and third, fifth and third, fifth and third, fifth and third. Can you understand that one? Fifth and third, fifth and third. Can you understand that one?
Yes, sir.
Can everyone understand it?
Yes, sir.
Good. If you so, if it is so, is this one, this fifth carbon, say the chain is finishing there, is this one involving to form the phosphodiester bond?
No.
No. This
No.
one is not making the phosphodiester bond. This one is also not making the, what is it? Phosphodiester bond. That place we call 5' end. How do you call it? How do you call it? 5' end. And this end is called 3' end. Understood? Understood. So what is meant by 5' end? They are fifth carbon stays without making phosphodiester bond. That is one end. Opposite end will be 3' end. What is the meaning? They are third carbon stays without making phosphodiester bond. Clear?
Clear.
Yes, sir.
Please mark the details. Completed. Completed.
Yes, sir.
Good. Now, my children, take this chain. If you take this chain, you tell me which end is this? Is it 3' or 5'? You decide.
Three prime.
It's a 3' end. Correct? Am I right? Do everyone agree?
Yes, sir.
Yes. Then the opposite end will be? The opposite
5'
end will be
5'.
5' end. So do you agree in the DNA molecule there are two chains, correct? There are two chains. If this is 5', if that is 5', then the other end is, what is it? 3'. Agree? Agree?
Yeah, sir.
Yes, sir.
Then this is, that side is 5 prime mean its opposite end is 3
3
prime. Then that side is 3 prime mean its opposite end is
5.
5 prime. This nature of DNA is explained to you by the word that they are, what is it? They are
Antiparallel.
anti-parallel. Could you please mark it down? Have you marked it?
Yeah, sir.
Right.
Yes, sir.
Here, one more thing. Can you see these dot lines? Can you see?
Yes, sir.
They are the home
Hydrogen bonds.
hydrogen bonds. Okay. So, between adenine and thymine, okay, remember adenine is always a pair to thymine. Wherever adenine there, in the opposite side, who will be there?
Thymine.
Thymine. Wherever guanine there, in the opposite side, who will be there?
Cytosine.
Cytosine. So adenine is a pair to
Thymine.
thymine. Guanine is a pair to
Cytosine.
Hydrogen.
cytosine. Adenine and thymine are connected by how many hydrogen bonds?
2 hydrogen bonds.
2 hydrogen bonds. Guanine and cytosine are connected by
3 hydrogen bonds.
3 hydrogen bonds. Clear?
Yes, sir.
Right. In that case, so could you please mark it down the hydrogen bond? Okay. Now, if you take this one like this, there are pairs, correct? It is called a base pair. How do you call it? How do you call it?
Base pair.
It's a base pair. It is a base pair. Mark the base pair. Marked it.
Done, sir.
Right. Finally, look at this. If I take like this, what are the components coming here? Nitrogen bases are excluded, correct? Am I right?
Yes, sir.
Right. This component is called sugar phosphate backbone. What is the name given? What is the name
Sugar
given?
phosphate backbone.
Sugar phosphate backbone. So sugar phosphate, sugar phosphate, diester, phosphodiester bond. Sugar phosphate, phosphodiester bond. Sugar phosphate, phosphodiester bond. This arrangement, how do you call it? Sugar phosphate backbone. Have you marked it?
Yes, sir.
Right. So this idea is the one written here. It is a, what is it? Double helix structure. How many complementary strands are there? Two strands. Okay. These two strands are arranged in what form? In what form? Spiral. Correct? Helix means spiral. Helix mean, what is the meaning?
Spiral.
Spiral. Wait a minute. Right? Next it is speaking about the anti-parallel nature. Anti-parallel nature. Do you agree with the anti-parallel nature?
Yeah.
Yeah. Sugar phosphate backbones are present outside of the helix and the center is taken by whom?
Nitrogen bases.
Nitrogen bases. Who is in the outside?
Sugar phosphate backbone.
Sugar phosphate backbone is making the outside of the helix. The center part is made by
Nitrogen
center
bases.
part is made by
Nitrogen bases.
nitrogen bases. Further, the two strands are held together by
Hydrogen bond.
hydrogen bonds. The deoxyribonucleotides are connected through
Phosphodiester bond.
phosphodiester bond. They are long and linear. If they say linear, what is the meaning? If they say linear, my children, no branches. Understood? No, what is it?
Branches.
Branches. Okay. Each nucleotide contains a phosphate group, deoxyribose sugar molecule, nitrogen base. Two types of nitrogen bases are there. What are the two types of nitrogen bases? Purines and pyrimidines. What are the purines found in the DNA?
Adenine and guanine.
Adenine and guanine. What are the pyrimidine nitrogen bases found in the DNA?
Cytosine.
Cytosine and thymine. Are you clear?
Clear, sir.
Right. What is the next? So please go through the content. Please go through this content and tell me whether you are clear with all the points. Have I explained you all the points? Are you clear?
Sir,
Yes, Putta.
won't the uracil come for pyrimidines?
Ama,
Won't the uracil come for the pyrimidines?
is uracil there in the DNA? Do you have uracil in the DNA? Are there uracil containing deoxyribonucleotides?
No, sir.
We have pointed right here. Ribo only uracil is present, correct? Deoxyribo mean to that place who is present.
Thymine.
Thymine. So is there any opportunity to be uracil in the DNA?
No, sir.
Is there any opportunity to be thymine in the RNA?
No.
No. Three will be common. Adenine, guanine, cytosine will be common for both. Clear? So here what we are writing is purines in the DNA. Here what we are writing is pyrimidines in the DNA. We are not writing common purines and pyrimidines. Understood?
Yes, sir.
Right. What is, so is it clear? Rest of the components are clear, my children?
Clear, sir.
Clear. Now what is the heading? Base pair rule in DNA. Okay. General, oh no, if you are asked in the exam, what is base pair rule? What is base pair rule? You have to write this. Always a purine base pairs with a specific pyrimidine base. What will you write? When in the exam paper, they will ask you, what is base pair rule? What is base pair rule? What is base pair rule?
A purine base pairing with a
Always,
pyrimidine base.
always a purine base pairs with a
Pyrimidine base.
pairs with a
Specific pyrimidine.
specific pyrimidine base. Mark it down, write it down. It's already written down. Can you see? It's already written here. You can just take it up. But thing it is, that should be taken as the home
Definition.
definition. In exam paper, if they ask you what is base pair rule or define base pair rule mean, you have to say always a purine base pairs with a specific pyrimidine base. Clear?
Clear, sir.
Have you marked it?
Yes,
Right.
sir.
Here you can see the base pairing. Base pairing between whom and whom given here?
Adenine and thymine.
Adenine and thymine. Between adenine and thymine, how many hydrogen bonds are present?
Two
Two
bonds.
hydrogen bonds.
Two. So we mark a 2-line bond and mark T. Mark it down like that on the right side or left side. So how many hydrogen bonds?
Two hydrogen bonds.
Then how many lines you mark?
Two lines.
2 lines. Here, guanine and cytosine, how many hydrogen
Three
bonds are there?
hydrogen bonds.
Then how will you mark? How many lines you will mark?
3 lines.
3 lines and mark it. I can't mark it here in the typing. Triple bond will not come. Have you marked it?
Yes, sir.
Have you marked it? Are you okay with this statement? Are you okay with that statement?
Yes, sir.
Yes. Do you agree always DNA shows anti-parallelism?
Yes.
Yes. And this is a new point to you. In every turn, one complete turn in the DNA has how many nitrogen bases? How many nitrogen bases?
10 pairs of nitrogen
10
bases.
pairs mean how many nitrogen bases?
20.
20. So 20 occurring in 10 pairs. Clear?
Yes, sir.
20 occurring in, what is it? 10 pairs. That is how to understand. Okay, now coming to the heading, functions of DNA. What are the functions of DNA given? Read this one.
Stores genetic information as a sequence of nitrogen bases and transmit it from one generation to next generation.
So, genetic information is stored in what form? What form? Sequence of nitrogen bases. And it is transmitted from one generation to another generation. Okay. What is the second function?
Stores the genetic code for protein synthesis.
Right. Code is okay, but you correct, code is okay, it's not a wrong word. Code is okay. Code is, you convert it to genetic information. Genetic, what is it? Information. So, okay, convert genetic information for, what is it?
Protein synthesis.
Protein synthesis. Remember, these two are the two main functions directly given to us in the syllabus. Understood? These two functions are the ones direct in syllabus. Direct in syllabus. So what are the two functions of DNA? What are the two functions of DNA? In exam paper, say they are asking what are the two functions of DNA? What will you write?
So genetic information as a sequence of nitrogen bases and transmit it from one generation to another. Stores the genetic code for protein synthesis.
Genetic information. Store genetic information for, what is it?
Protein synthesis.
Protein synthesis. Another four are there. They are in the syllabus, but can't write giving priority to the first and second. Understood? Providing code for protein synthesis, controlling cellular activities, making the primary structure of chromosomes, self-replicating and forming identical copies. They are functions of DNA. They are in the syllabus. Thing it is, in the exam paper, if they are asking, write two functions of DNA mean, which one will you write? These two.
The first two.
The first two. Okay. We are not studying here the things for the knowledge. We are studying it for the exam. Clear? We are studying for the exam. If you write this, you will get marks. If you write this, you will get marks. If you write this, you will get marks. That's all we have. Understood?
Yes, sir.
Right. So that's what I'm telling. So which is direct in syllabus. Direct in syllabus, write that one as the first priority. Clear?
Clear, sir.
Good. So what are the places you can find DNA in a cell? Read this one.
Nucleoid,
Nucleoid is a region. Okay. What is nucleoid? Nucleoid is a region in the cytoplasm of prokaryotic cell that contains, okay, central circular DNA. What kind of DNA? What kind of DNA, my children?
central circular DNA.
Central circular DNA. So please write it down this. Wrote it?
Yes, sir.
Wrote it, my children? Okay. Next it is plasmid. Plasmid, okay, plasmids are small circular DNA molecules. What kind of DNA molecules? Okay, that carry few genes. Plasmid are small circular DNA molecules that carry what is it?
Few genes.
Few genes. Okay, you wrote it. Have you written it?
Yes,
Have
sir.
you written it? Good. So look at this diagram. Can you see this diagram?
Yes, sir.
This is what we call circular DNA. If it is a circular DNA, there will be anti-parallel nature. But circle no baba. This is 3' end. This is 5' end. Can we identify?
No.
No.
Count.
Circular DNA we can't identify. They have the anti-parallel nature. In one place, if you cut it, one line will be 3' to 5', other end will be 5' to 3'. That will be there. This is what kind of DNA molecule? What kind of DNA molecule?
Circular DNA molecule.
Circular DNA molecule. Okay, that is one thing that you want to know. It's a circular DNA molecule. I'm sharing this picture in the group. Okay, just for your idea, right? Normally, DNA mean it is called what kind of DNA? What kind of
Linear
DNA?
DNA.
Linear DNA. Linear DNA mean it will be like this. Can you see? So suppose if this end is 3', its other end will be
5'.
5'. And oppositely, this end will be
5'.
5'. And the other end will be
3'.
3'. That is linear DNA. So what are the two types of DNA? What are the
Circular
two
DNA.
types of DNA?
And linear DNA.
Right. One more thing, right? Here we are using a word called nucleoid. We are using a word called, what is it? Nucleoid. So if you take nucleoid in a bacteria, in its center, there's a region. That region is called by the name, what is it?
Nucleoid.
Nucleoid. In that nucleoid, the bacteria's main DNA will be placed. The bacteria's main DNA will be placed in the
Nucleoid.
nucleoid. Okay. Additionally, there will be small DNA. They are the plasmid DNA. In a eukaryotic cell, what are the places you will, eukaryotic plant cell, what are the places you will notice the DNA?
Nucleus, mitochondria, chloroplast.
Right. In the nucleus, what kind of DNA is there?
Linear DNA.
Linear DNA. In the mitochondria and chloroplast, there will be circular DNA. What kind of DNA?
Circular
What
DNA.
kind of DNA? Circular DNA. Mark it down. Bracket mitochondria and chloroplast and say what kind of DNA? Okay. Completed.
Yes, sir.
In animal cells, where will you find DNA?
Nucleus and mitochondria.
What kind of DNA will be in the nucleus?
Linear DNA,
Mitochondria?
circular DNA.
Right. So before I enter to the ribonucleic acid, some ideas with you. One, are you collecting the terms continuously?
Yes, sir.
Who are the ones collecting the terms? Term collection. Tell your name. Who are the ones collecting the terms? Rima, you are collecting the terms.
Yes, sir.
Amna,
Yes, sir.
Anam,
Yes, sir.
Azim is today new to our class. I will inform Azim separately. And Asma, last class only joined us.
Yeah, what term, sir?
In this class, Baba, children, tell me what are the books you are maintaining?
Term collection, definition, essay, short questions.
You have to maintain how many books? Asma?
4.
Term collection mean every class you study, you will come across certain terms, correct?
Yes, sir.
Nucleoid today, circular DNA today, correct? Anti-parallel today. How many words we study today? Collecting them is term collection. Definition mean for all the words are we giving definition?
Sir, excuse me, sir.
For all the words, do we give definition?
No.
No. The definition you are given, you have to collect. Short questions, I will send. You have to answer. What is the last short questions you people did? Arima? Carbohydrates done. Arima?
No, sir.
Then what is the last? Water? Did I give water?
Yes.
Water was the last one, teacher. Sir.
Inshallah, I will give you carbohydrates, lipids, and proteins this week. Okay.
Okay, sir.
Excuse me, sir.
Yeah.
Can I have the previous notes and stuff for these things, the photos of the...
Inshallah, you will get it. Don't worry. Okay.
Yeah, okay, sir. Thank you, sir.
If I promise, I will give anyway. Okay. Inshallah. Don't worry. கொஞ்சம் முன்னுக்கு பின்னுக்கு ஆகும் பட் கிடைக்கும்னா கிடைக்கும். Okay.
Okay, sir.
So your empty book is which book?
Essay question.
Essay. Can't keep that one empty, no. Correct. So the first heading, I am going to give you to write an essay. Remember, when you are writing essay, at the moment, now that essay, when you read the heading, you will understand which area you are covering, correct? You have to well prepare yourself and write the essay. Understood? Understood?
Yes, sir.
So, this is the heading. Describe the properties of water that contribute to maintain life on Earth. Describe the properties of water that contribute to maintain, what is it?
Life on Earth.
Life on Earth. Today is Tuesday. Before Friday night, can you complete this essay?
Yes, sir.
Before Friday night. Inshallah, Friday night, you write the essay. Before Friday night, you write the essay. Before Friday night, you send it to me privately. Or in the group also, okay. But normally A-level children are sending me privately. Okay. Normally, Rima has to send Rima's work. Anam has to send Anam's work. Understood?
Yes, sir.
No need to send in the group. Friday night, for every child who is sending the work, I will distribute the scheme. Okay? I will distribute the scheme. You have to correct it. If possible, I can't force that one, but better choice it is taking a printout of that scheme and attaching that one to your essay book. Clear?
Clear, sir.
We have a basic rule here. Remember, in the essay, we score out of 150. If a child is getting 150 out of 150, perfect. Clear? Here, 145 to 150, it is first place. 140 to 145, that is, what is it? Second place. 130 to 140, that is, which one? Third place. If you are scoring less than 130, then you need to rewrite the essay and again score it. Understood?
Yes, sir.
This rule we are strongly following. Remember, these things we do to improve your performance. Understood? We do these things to improve your performance. One of the key character a student needs to get best in the advanced level, it is, okay, should not feel, what is it? Should not feel lazy. Should not be, what is it? Careless. Okay. Should not postpone work. Okay. Due to any reason. Reasons can be many, but should not, what is it? Postpone the works. Okay. If a person, now for any person's success, these things are important, right?
Careless.
For any person's success.
Careless.
என்ன சத்தம் கொண்டு வருது? Okay. So you are in the very beginning of this process. First essay I have given, yet I have not given you any exams. Did I give you any exams?
No, sir.
No. So coming Tuesday, inshallah, you do an examination on carbohydrates. Okay. In whom? In whom?
Carbohydrates.
Carbohydrates. We'll see. Okay, so keep this in the mind. I have given the first heading to you. Coming week, I will give you the next heading. Okay, if on Friday night, I will release the scheme of it. I will release the scheme of it. Okay, you have to correct your one and send me the score. If I notice your score is less than 130, I will ask you to rewrite it. Then you have to rewrite it and send that score also to me. Until I notice you are getting above 140. 140 above means second place, right? I will request that. Keep in the mind. Okay. So with those ideas, we are coming to the heading called, what is it? Ribonucleic acids, RNA. If you take RNA are polymeric molecules made by the polymerization of ribonucleotides attached by phosphodiester bond. Here, uracil, cytosine, guanine are there. Remember, in the RNA, if a base pairing occurring, okay, remember, in DNA, always there will be base pairing. In DNA, always there will be, what is it? Base pair. But in RNA, okay, in RNA, some RNA molecules only show, what is it? Only show base pairing. So do you notice base pairing in RNA always? Tell me. Do you notice base pairing in RNA always?
No.
No. Right? In that place, adenine will be paired to whom? Adenine will be paired to whom?
Uracil.
Uracil. Guanine will be as usual paired to whom?
Cytosine.
How many bonds will be there between guanine and cytosine?
3 bonds.
3 bonds.
3 bonds. Complete it. Completed?
Yes, sir.
In the RNA, there are 3 types of RNA. Read this name.
Messenger RNA,
Short form.
mRNA,
Second one,
transfer RNA,
short form.
tRNA,
Third one,
ribosomal RNA, rRNA.
rRNA. Remember, if you take mRNA, in mRNA, no base pairing will be there. It's a linear structure. One base and another base are not coming face to face. Clear?
Clear, sir.
Mark it down. Right. If you take tRNA, it has base pairing. You can see here. It has, what is it? Base pairing. rRNA also has base pairing. Understood?
Yes, sir.
Right. Actually, it's a small portion to complete this portion, but we have to discuss certain important points there. Cellular basis of life tube, I will send you. Take the printouts and stay ready. Okay.
Okay, sir.
Okay. Okay. We'll meet inshallah. Assalamu alaikum wa rahmatullahi wa barakatuh.